So I'm Clifford Rostomily. I am a second-year graduate in the Trapnell Lab here at Genome Sciences. And I'm excited to kind of tell you guys a bit of genome adjacent stuff. So the genome isn't just composed of DNA in your cells. There's actually hundreds of proteins that are interacting with the genome. You can, like, collectively, these proteins are all called epigenetic regulators. And you can appreciate just from this picture, this is from a epigenetic regulator database that, you know, there's tons of diverse functions and they all interact in this very complicated network. Now, an interesting thing about these epigenetic regulators is that a large number of them are associated with developmental disorders. And this kind of makes sense. They're ubiquitously expressed and they're really required for precise gene expression and development. And you can see that the number associated with developmental disorders has just been increasing over time as we've been able to sequence more human genomes and link variants in these genes to phenotypes and disease. And so currently there's 85 epigenetic regulators associated with developmental phenotypes. And this encompasses roughly 30%. And I'm sure that over time, this number is just going to increase as we start sequencing more genomes. Another striking thing about a lot of these regulators is that when they're mutated, they have not exactly, like, you can still clinically discern the phenotypes, but they kind of affect the same processes in development. So one major feature of a lot of these epigenetic regulators is that you'll see kind of craniofacial just like defects in development and cognitive impairment and kind of musculoskeletal defects. So I've been really interested in why all these epigenetic regulators converge or like have like certain cell types have similar susceptibility to mutations. And so these are just kind of some of the common phenotypes you see: intellectual disabilities, growth abnormalities, and neurodevelopmental syndromes. And so to study these regulators, I've been interested, like you kind of have to pick a model, right? And this is the model organism section. And so if you think about modeling, you know, I just told you there's a ton of these regulators. Modeling them in mice, well, that's a great choice because they're mammals, so they're kind of closely related to humans in a way. However, mice are really hard to work with. You know, the embryo develops within the womb of the mouse, and so you can't directly see it, and they don't have a ton of offspring. On the other end of the spectrum, Drosophila, or the fruit fly, has a ton of offspring. You can do all kinds of mutagenic screening and really cool genetic work in them, but they're obviously pretty different. So in our lab, we work with kind of the middle ground of these two, the zebrafish, Daniario. And zebrafish are great because they have a ton of offspring and they're optically clear and they grow pretty fast. And so just to kind of give you a flavor of this, this is the first 24 hours of zebrafish development. And you can see that the embryo here is encased in this layer, which is called the chorion. This is a protein layer that's permeable to nutrients or chemicals, and so you can chemically treat the embryos. And this whole movie will be 24 hours. So by the end of this, you'll be able to see that you can already see kind of like a, you know, semblance of a fish. And at 24 hours, they're already moving around a bit. And by 48 hours, they can freely swim. And this is just one egg. In a typical experiment, we can generate upwards of 200 embryos to do work on. And so with the model system in place, I wanted to kind of start going after some of these epigenetic regulators. So I've first been targeting this protein KMT2C, or its ortholog KMT2D. And this protein has kind of complicated functions that are not very well known in the genome. And I won't go into all the details here, but you can see that it acts as this massive scaffold for a ton of other proteins and is involved in a bunch of interactions that are important in specifying gene expression. And when it's mutated in humans, it results in these two syndromes, Kabuki type 1 and Kleefscher syndrome type 2. They're characterized by these craniofacial dysmorphisms, short stature, cardiac defects, among other things. There's a huge list of symptoms in these patients, and they have very challenging care. So it's important to kind of know the underpinnings of this disease and why this protein KMT2C is causing their phenotypes. And so to study this, I'm employing an approach that was just alluded to, CRISPR. So what CRISPR is doing is we're designing these RNAs these proteins complex with RNAs that can target a very specific part of the genome and make a cut or an edit. And so I just have this picture on the left of normal DNA, and on the right, it's the mutations induced by CRISPR targeting. And so in reality, it's a little more complicated, but all you need to know is that I can knock out these genes in an individual embryo. And the way we deliver these CRISPR reagents is we micro-inject into the zebrafish embryos. So we pull a very fine needle, and then we just poke it and shoot a little bit of nanoliters of reagent into the embryo with a kind of nitrogen pump. And then we grow them up and we compare the wild type. And so what I'm showing you here is a stain of cartilage. And so in the developing embryo cartilage, you get cartilage before you get bone. So this is kind of where the bone's going to be in the final embryo. And you can see in the embryos where we've targeted KMT2C and D with CRISPR, we have defective craniofacial cartilage patterning. And this is pretty repeatable. Another study that made a pure knockout, so they didn't just inject in the one cell stage, they have every cell knocked out for KMT2, has also shown a lot of cartilage defects. And so what's great about this is that we can model these diseases that are occurring in a small number of people. And so there's not a lot of funding to build a whole mouse and a whole grant around one of them. And we can model them rapidly by just targeting these genes with CRISPR. And so in order to, this is great and all, but in order to figure out what's going on in them, we employ this assay called single cell RNA sequencing. And so just a little bit of a primer on what that is. I don't know if many people have heard of single cell RNA sequencing, but you can think of single cell RNA sequencing as, instead of taking a whole embryo and just grinding it up and then looking at all the RNA that's present, which would be kind of amalgamation of all of the cell types in the embryo, we take a zebrafish, we dissociate it into its individual cells, and then we sequence the RNA present in each of those. And what that tells us is the cell types that are present in the embryo and their abundance, and the genes that are being expressed or perturbed in the knockout embryo. And so a basic experiment here would be we inject the CRISPR-Cas9 reagents targeting the KMTs. I screen for knockout and control embryos, and then I perform single cell RNA sequencing. I don't know, there's a sky there because the method is actually called sky RNA sequencing, but I took that out and did not change the sky. And so what we get are 400,000 cells encompassing 173 distinct cell types. And so this really lets us look at each cell within the embryo and how it's affected by knocking out these genes. And it's a really powerful way to phenotype and kind of get down to what's going on. And so I'm just going to give you kind of one vignette of what we've seen in this. And so in the eye, right, when you get exposed to light, the light comes through your eye and actually hits the back of your eyeball. And it triggers it gets detected by these rod and cone photoreceptors, which then fire and send a signal back up through an optic nerve fiber. So the correct development of these neurons is really important for vision. And in Kabuki and Kleefscher syndrome, there's a lot of visual defects in the patients. And so in our zebrafish, we actually see we can actually pull out all the cells that are involved in neuronal differentiation. And we can look at, you can see here, I've labeled them. So I've plotted these on, this is called a UMAP. It's just like a way of taking all the high dimensions of gene expression and compressing them down into 2D space. And so you can see in this cluster here, there's the progenitor neurons. These then differentiate into these precursors, which then get fully differentiated into the retinal neurons that you kind of see over here. And so we can track this whole process when we're sequencing zebrafish. And then we can look at the difference or change in abundance of these cell types in the wild type versus the edited embryos. And what we can see is that a lot of these neurons are getting stuck in a progenitor fate, so they're not able to fully specify into these cell types. And then one thing I forgot to mention is that all of these neurons come from a single cell type that then receives signals and causes them to differentiate into each of these. And so we see this as kind of blocked, and the progenitors are kind of increased, and they're not able to differentiate down into the terminal cell fates. And so we wanted to then validate this. So we have a method of staining for gene expression in the zebrafish eye. And so you can see right here, this is like what I'm taking a microscopic image of. And what you see is that when I stain for the genes that mark the mature photoreceptor neurons, for instance, here, that in the knockout, they're completely absent, which is indicated by the loss of the blue stain. But in the wild type, they're there. So this method's really letting us pick out all the kind of cell systems that are affected by knockout of these epigenetic regulators. And the future directions will be to really nail down the mechanism of why this process is interrupted. And yeah, I didn't put a slide for that, but I just want to acknowledge everyone in the lab, this Trapno lab, especially Amy and David, who I worked with.